people are only a couple minutes away, so we'll start with the introduction, and then we can get going, and he'll be here by the time we get. So this is the dissertation, our thesis, dissertation defense for Steve Quistad. I'll never say that. And this is how I first met him. Is isn't he cute? So he's basically always been an overachiever as far as I can tell. So Dave Valentine from UCSB wrote me and said, look, we have this student, he has his own funding and he wants to study coral viruses. And he had been planning to go off to Australia to do this work, but then something fell through with the funding and everything. So he came here to start doing this, and he, when you go and look him up, he's like this, highest honors sort of thing in some weird UCSB thing with president and founder of this organization, blah, 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 and and he smiles, and as everybody knows, he's always smiling, right? So in my lab, we of course try to crush any happiness out of your life as we go through time. So there he is smiling, and then we have the Texan, right? And usually he does the full metal jacket thing, and most everything crushes under just this part of it here. But he's still smiling. And then we went with the unreasonable workload. So this is him carrying several hundred pounds of ice off the glacier, filtering it. I was helping a lot right here. And a whole bunch of other things where we send him around the world. And he works like a dog, so to speak. Then I tried to kill him, sorry about that. Slight miscalculation. It was only a miscalculation that I was there. I would have been So he's still alive, and he still smiles. So there he is standing, we're talking, this is like -2 degrees, and he's like, haha, that's great. So then we thought, well, if we can't do this, why don't we just go to can we just corrupt him, right? And so when you're going to corrupt someone, of course, you just get Canadians and Australians. And so this so it does work, right? So this is a Canadian, which is really about as bad as it gets. This is actually even worse. This is a Canadian Australian, which is like the worst thing ever. And of course, these guys have been hanging around a whole bunch, and this is kind of what happens.
Oh my God.
Sorry, maybe you guys should close your eyes in the third row there. You right? So this is weird things with the koala. Horrible. That's the dress code, that's what happens to you. And then they, there are so many things wrong in these pictures. All right, and then despite all of this, and Steve had a great addition to the lab, and we've done tons of things together, and it's been a lot of fun. 3 published papers, 1 that's in revision right now, and 4 that are mostly written, and then he'll be sending out between now and November, is the idea. He's done a lot of outreach, actually. So he goes to high schools and talks about things and so forth, and does things with the museum, what is it? The Aerospace Museum, the 2 science and art things and so forth. He's also got some of that's the highest awards that you can get at SDSU, the ARCS and the Provost Award. And then he got a postdoc with Dr. Paul Rainey, who's literally one of the world's best microbial evolutionists. And he'll be going to Paris in November, is that right? So that's a super cool thing. And he's still smiling despite all of this. This is actually my favorite Steve picture ever. So this was us in Franz Josef Land, and this guy has swum to the middle of the lake, And they had gathered this little sample, and then they were looking off, trying to look like explorers. This is how I always remember it. And then the last thing, of course, before we let Steve go, is that there will be a party on Saturday for everybody that wants to come. So 3 to 5 will be at the S&M restaurant on Park, and then 5 will be at Denai's house for drinks and stuff. And with that, I will let Steve take over. No. Fail.
Okay, thank you very much for that introduction for us. That was a lot better than I was expecting after 5 years in your lab. So today I'm really excited to tell you guys all about what I've been up to for the last 5 years, where I've been exploring viruses in corals and what that can tell us about where animal life came from on the Earth, so animals being metazoans. I'd like to start off by thanking my committee members, Dr. Rob Edwards, Dr. Golan Volkovitz, and the chair Forrest Rower, as well as Dr. Martin Trescaris, who is on his way over from Scripps right now, and Dr. Stephen Hedrick from UCSD, who couldn't make it today because he had a car emergency this morning. So with that, getting into my talk, so you see in the title of my talk, I have this origins word. So I thought it'd be appropriate to start with putting everything into context of life on this Earth. So this is really my favorite analogy to really comprehend how long the Earth has been around, how long life has been around, and actually how quickly some of these major evolutionary transitions can happen. So if we condense 4.6 billion years into a single 24-hour period, starting at 12 a.m., down here we have billions of years ago from 4.6, 3, 2, and 1. This is what the history of life looks like. So about midnight we had the formation of the Earth. About 5 hours later we had the first prokaryotic life appear. Another hour went by, we had the first photosynthetic prokaryotes. These were the first guys to fix carbon dioxide, releasing oxygen, causing this thing called the oxygen revolution, one of the major evolutionary transitions. Then almost 12 hours later, at 5:11 p.m., we had the emergence of the first eukaryotic organisms. Next, about an hour and 50 minutes later, we had the first multicellular life. Probably looks something like a group of cyanoflagellates decided to hang around together. Then at 9 p.m., only 3 minutes until midnight, we had the first animals. And to the picture I have here is a coral. That's what you'll be hearing about today. And they're arguably the first animals out there. Then at 11:41 p.m., only 19 minutes until midnight, we had our first primate ancestors emerge. Then at only 0.08 seconds until midnight, we had the first modern humans. And then finally, this last major event, which I know is important to a lot of people in this room, at only 0.002 seconds until midnight, we had the first beer that was brewed. And so while this did affect my dissertation work, what I'll be focusing on today is this event that happened from about 9 until 9:15, representing about 50 million years of evolution. And this event is referred to the Cambrian explosion. So explosion, that's quite a dramatic word that you don't usually hear a lot in biology. Why do we call it that? Well, it was during this relatively short period of time, 15 minutes or 50 million years, life on Earth went looking like something like this, a bunch of single-celled organisms, to looking something like this. Vast diversity of life all of a sudden appears in the fossil record. So all modern-day phyla that are around today, all their ancestors began during this very short period of time. And to show that, I have just a bunch of pictures of representative invertebrates that I've taken through my fieldwork. And so what you'll be hearing about today is one of these specific animals that emerged during the Cambrian explosion, reef-building corals. So why am I talking about corals? So corals are members of phyla Cnidaria, which is an ancient metazoan phyla. It diverged from Bilateria, which includes everything from worms to humans, anything with bilateral symmetry, about 500 million years ago during the time of the Cambrian explosion. So you saw in my talk, I have viruses. What do viruses have to do with corals? Well, viruses are really the major drivers of evolution on this Earth. And just to kind of exemplify this, I have this graph here that looks at the mutation rate per base pair per generation, taken from a paper in 1998 by Drake et al. And we have down here this small little blue line, that's humans, about 1 × 10⁻⁷ mutations per generation. So this guy right here in the green, that's their RNA viruses. So you see here the y-axis is broken because I had to change the entire scale where the viruses are mutating at 4 to 5 orders of magnitude more than we are. So you have one generation, you have a couple kids, that's a couple copies of your genome. Well, viruses can have anywhere from 10 to 10,000 viruses in a single generation. So they're mutating faster, and there's more of them. And the most important aspect is that viruses are these things that we call obligate intracellular parasites. And that's just a fancy way of saying that a virus needs a host to make more viruses. They can't make a new virus all by themselves. And this is the most crucial fact here, because by the very nature of how viruses are required to exist, we have viruses that are mutating, there's a lot of them, and they have to interact with the host to survive. And it's these interactions that really drive host biology and drive evolution. And whether you're in a microbial system or eukaryotic, the viruses are basically in charge. So a basic outline of where I'll be taking you today. So the overall purpose of my dissertation work was to understand how viruses have driven metazoan evolution from the Cambrian explosion to present day. And to do this, I use corals as a representative basal metazoan organism. And these 3 chapters represent the 3 core chapters, which are independent papers that have come out of my dissertation work. So the first story I'll be telling you about are viral interactions with the host immune system, and how we can actually use viral populations to predict how unknown immune systems are structured. And this is in preparation, hopefully submitted in the next couple weeks. Chapter 3, looking at how viruses interact with host genomes. And I'll be telling you a story where I identified, to my knowledge, the oldest retroviral infection that's ever been described in the literature that occurred during the Cambrian explosion, with implication for how cnidarians develop their body plans. And this is currently under revised submission at PNAS. And then the 4th chapter, I'll be looking at the evolution of a specific host antiviral response called apoptosis. And this was published in PNAS in 2014. So just to quickly look at the products of the dissertation. So these are in a variety of formats, published, submitted, in prep. And these are the core papers that have come out from specifically my dissertation work. And then these are other projects I've been involved with that weren't directly related to my project. So what was known about viruses in coral before I began this 5-year journey into coral viruses? So this paper by Davey and Patton, published in 2007, looked at the morphological diversity using some TEM. And so here we see some really cool images of phage VLPs. So phage are the viruses that infect bacteria. And why we say VLPs, that stands for virus-like particles. So in environmental virology, and specifically with corals, we haven't been able to isolate a virus that we can infect in coral and get that same virus out. So we have to call them virus-like particles because they're the same density as known viruses, and they stain with nucleic acid stains. So throughout this talk, if you see VLPs, just think viruses. We also have some really interesting polyhedral ones and some cool filamentous ones. This guy F, this lemon-shaped one, that looks like it's probably associated with an archaea that's associated with coral. So we know they have a diverse population. How about functionally? What's known about coral viruses? So some work done by Dr. Becky Vega-Thurber in 2008. She looked at physiological stress and how that affects specific types of viruses, looking at these herpes-like viruses that came out through her metagenomics. And she found that when you stress out a coral, you have more herpes-like viruses. And this is kind of the core piece of data that came from that paper, where we have time and hours, and we have copies of a specific sequence from that herpes virus. And this was some qPCR data. And so I just want to draw your attention to the temperature, which is this red line. And we see that when you heat up the coral, you have more copies of this herpes-like viral sequence, which is a proxy for herpes virus abundance. So you stress out a coral, the herpes viruses pop out. What's really cool about this work is it got some interesting press, which is really at the pinnacle of pop culture and science. So it was covered in the Colbert Report in 2007, and he featured it on his threat down for coral reefs. So this article, covered by Yahoo, herpes viruses are apparently killing the coral reefs. And his defining quote from this segment is, "This leads to one obvious question: who has been effing the coral reefs?" So pretty cool to see coral viruses on such a national and international scale. So basically, before this dissertation work, we knew corals had diverse viral populations based on morphology and based on sequence data. We knew that specific viruses, those herpes ones, change in response to stress. However, interactions between specific virus-host components were completely unknown in the coral system. And that's the focus of my dissertation work. So The first chapter I'll be telling you about is looking at how viruses interact with the host immunity and how we can actually use the viruses as predictive tools to better understand how the host immune system is set up. And this work is in preparation, hopefully submitted in the next couple weeks. So how do we look at viral-host interactions in traditional systems? So what we have here, we have mice, fruit flies, and worms, some of your traditional model organisms. And four main tools that we have at our disposal in these systems are antibodies, which allow us to look at specific proteins, tissue culture, where we can actually culture specific cell types, see how a virus interacts with those cell types, and then the ability to knock in and knock out genes to see any loss of function and for phenotype and what that gene might actually be doing. So what do we have in core when we come into the system? We have none of these things. So we have no antibodies that we can just go on to Santa Cruz Biotechnology, order, have them come the next day and start our experiments. Tissue culture is kind of an interesting one. So there's about 5 papers that have been published on how to culture coral cells in the last 15 years. None of them have been cited. They only cite each other. And I spent about 4 months of my life trying to reproduce them, and it didn't work. So for all intents and purposes, we don't have the ability to culture coral cells beyond a couple weeks. And then we have no ability, there's also a paper that's come out where they tried doing corals to knock in all the genes. Once again, not citable, not reproducible. So basically, we have none of the traditional tools that most people use to study how viruses interact with their hosts. So what we need is a broad tool that we can apply to not just coral, but if you remember that image from the Cambrian explosion, to develop antibodies, tissue culture, gene knockout, knock-in. We spent 4 decades getting these guys going. We're not going to be able to apply this to the entire tree of life. So what we really need to get at how all of these unknown immune systems are set up is some broad tool that we can apply to non-traditional systems. And that tool is what I'm proposing is using viral populations to predict how host immune systems are actually structured. So what do I mean by that? So if you remember earlier I said viruses are those obligate intracellular parasites. They need a host cell to survive. So a virus gets in the host cell, the host wants to kill that virus. And so we have basically 3 groups of proteins where we go from one virus to another and how you make a new virus. There are these first 2 which are basically involved in just making a new virus particle, those that replicate the genome, those that synthesize viral proteins. Then there's this third category which I'm most interested in that don't fit into these first 2, and by default we hypothesize that they're involved with evading the host immune system. So to kind of point out how viruses evade the host immune system, what I have here is this human herpes virus, the HHV8 genome, and this green bar basically represents 50% of its entire genome, and half of its genome is dedicated to just immunomodulation, not making a new virus, tricking the host so it can't get caught, and getting around all the host antiviral responses. And then just for some of you immunologists out here, they have some proteins with apoptosis, interferon chemokine, and these V's just refer to virus. You'll see some similar things, or some things you guys have probably heard of, GPCR receptors, IL-6, interferon factors, BCL2, etc. So viruses basically express these proteins that are similar to host proteins, but trick the host and allow for more viral production. So with this hypothesis that viral populations can actually be used as predictive tools, I predicted that if we compare all viral gene products to all host proteins, we should get 3 general classes of host homologs. Those being those first ones that replicate the virus genome. So I would expect those that are involved with DNA, RNA synthesis, nucleotide metabolism to pop up. Then those that are involved with synthesizing the viral protein, transcription, translation, protein processing. And then finally this third category, which I think is the most interesting and which I use to actually predict what the host immune system is. So basically the default is if we don't fall into these 2, we predict that it's involved with evading host immunity. So where did I get my core viruses from? So I'm doing some work down in French Polynesia and Moorea in the South Pacific. Here's the island of Moorea, kind of looks like a giant dinosaur footprint. And these stars here, that's where the research station is, and that's another public beach. I went down and collected them by scuba. The specific species of coral that I'm going to be telling you about is a Parides rus for you coral people. We pop open the tissue, we get the viruses out, we pop open their DNA and sequence it. And we get our viruses out. So the pipeline I use basically looks like something like this. So I have a coral virome, which represents 4 biological replicates, all combined into one FASTA file, making 500,000 sequences with an average base pair size about 250 base pairs. And then we have the coral proteome, which I compare it to, and they have about 24,000 proteins, about the same amount as humans, through this technique called BLAST-X, where I translate all the viral DNA into protein, match it up with the coral protein, and see and look for homologs. So from this analysis, you get two types of hits. You basically have those viral gene sequences that are related to a host protein, and then you have the host protein hits. So what I call a protein hit from the host had to have at least 5 unique viral gene segments matching to a specific protein with an e-value of less than 1 to the minus 4th. What I call a viral hit was basically anything that had 1 times e to the minus 4th compared to that protein. And then because this is DNA, we looked at some of their GC content, which I'll tell you about later. And then the protein hits, I tried to extract as much information out of them as I could using conserved domain databases and through BLAST-P comparing the human proteome to look for human homologs. So when I do it, if you remember, I have those 3 groups of proteins that I predicted I would find. So the first ones, those that are involved in making a new genome. So we have those nucleotide metabolism and DNA repair. So what these numbers represent, within the parentheses, that represents the number of unique viral gene segments. Sorry, no worries. That represents the number of unique viral gene segments that hit to 6 proteins. So 218 pieces of viral DNA, when translated, match to 6 different proteins that are within the general category of nucleotide metabolism. So first group that we expect, the genome replication. The next one, those that are involved with protein processing. So those guys pop up too, a bunch of hits to protein processing, translation, and transcription. Finally, the third category, which is the most interesting to me, those that are predicted to structure the immune system. And we see a bunch of proteins hitting that are known to be involved in immunity in a bunch of different systems. So down here we have basically the entire apoptotic signaling cascade. We have our TNF receptors, which you'll hear about a lot later. Interferon binding proteins, which are some of the classic antiviral responses. And then this RIG1, which is involved with nucleic acid sequencing. One important cool one to point out are these NLRs, or nod-like receptors. So corals have 500 of those compared to humans only have 50. They have the most nod-like receptors that have ever been seen. And they have many novel domain combinations that we've never seen in sequenced genomes. And so I have 413 viral gene segments that are hitting to 22 different nods. So that's pretty cool. So what I'm going to specifically be telling you a little bit more information are this RIG1, the TRAF6 and TRAF4, which are involved with TNF-induced apoptosis, which I'll tell you about later. And then this RET, which is proto-oncogene involved with cell cycle control. So basically at this point I have basically just homology that these proteins are similar. I wanted to provide further evidence that the sequences I'm looking at are viral in origin and not just host contamination. So to do that, I looked at the GC content. So the GC content of hosts is known to be different. The GC content of viruses is known to be different than their hosts. So I asked the question, these sequences that are matching to the protein, if we translate the gene segment from the host, translate the gene segment from the virus, and we compare the GC content, what does it look like? So we get something like this with these 4 representative proteins. So we have TRAF4, TRAF6, RET, and RIG1. And down below, we have the translated viral gene segment. On the top, you have the translated gene segment from the host with GC content here. So we have about 41 to 45 with the TRAF4, 46 to 49 with TRAF6, 46 to 29 with the RET, and about 47 to 63 with the RIG1. So you might be wondering then, well, so where are these viruses actually hitting these host proteins? Could they just be in a transmembrane region that's very conserved across a lot of different proteins? How do we know they're actually hitting to a part of the protein that's important for its function? So these regions are actually in those central domains. So where this virus is hitting this host protein is the part of the TRAF4 that is important for TNF receptor binding within the cytoplasm for signaling. With TRAF6, we have the TRAF-zinc finger domain, also important for docking intracellularly. Then with RET, this is the helicase C-terminal domain, and then the catalytic domain of the tyrosine kinase, which is part of RIG1. So these viruses aren't just hitting to a random part of the protein, they're hitting to the parts of the protein that are essential for function. So at this point, I have DNA evidence that I have similar virus proteins to similar host proteins. I wanted to ask the question, could I find any evidence at the transcriptional level? Are these things actually being expressed? So if you remember earlier with Dr. Becky Vega-Thurber's paper, I showed you that heat stress increases the populations of some viruses. So then I predicted that if there's more viruses hanging around in the cell, we should also see an upregulation of the host antiviral response. So to do this, I used some publicly available transcriptomes published by Steve Palumbi's lab in 2013. And they did this study in American Samoa where they took 16 acropora colonies, divided them into control and heat stress, did some RNA-seq, and in the end they had 482 differentially expressed genes, or DE genes. And so I hypothesized that predicted host immune genes from my virome analysis would also be differentially expressed under heat stress, with the prediction that because there's more viruses around, the host will be upregulating those antiviral genes. So when I took those genes that I found from my bioanalysis, went into their data set, they had already done all the statistics on it to show that it was differentially expressed. I could actually find 27 of those predicted genes that came up in the transcription. And these you can't read, you're not supposed to. I just want to point out a few. So we have things like complement activation, proteins that are involved with cellular proliferation. What you're looking up here is the full change in gene expression between the control and the heat stress. The really cool one is this zinc finger protein down here. So that was the second largest overall change in gene expression out of 482 genes. And that was predicted from my biome analysis. So there's this gene that the host is changing crazily that I was able to naively predict from just using my viruses. So this is the part I'm most excited for this pipeline, however, and that's the discovery of new types of immune components in a variety of systems. And so what I mean by that is all the data I've shown you at this point is based on domains that we know, that we've annotated, that we've shown in tissue culture that they do these immune things. So what about all that immunology out there that we're missing? How can we get at that immunological dark matter? And this type of naive approach, I propose, is one such way. So what you're looking at here, so these are two proteins, poor proteins with no known homology to any characterized proteins in all of NCBI. However, we can still predict where their transmembrane regions might be and whether their tails of their proteins are, the N and C terminals are intracellular or extracellular. And the stars that you're looking at, those represent 5 unique viral gene segments that are mapping to this specific region of the protein. So you can think of this as a recruitment map for virus gene segments matching to the protein. And what you'll notice is most of these stars are intracellular. So this makes perfect sense from a virus's perspective. If you get into a host and you have to manipulate your host cell biology, you do so from inside the cell instead of outside. And so this kind of approach, I think that we can find these proteins, we have no idea what they're actually doing, and then go into more characterized systems to test their function. So kind of expanding this, I propose a general pipeline to characterize unknown immune systems, which can be applied to essentially any animal out there. So you take your organism, you isolate your viruses, you do your transcriptome, you do sequencing of those viruses, RNA, DNA, whatever you want. With that transcriptome, you translate it, make a predicted proteome. You do BLASTX analysis comparing the viruses to the host proteins. And then you get those viral homologs in DNA and those predicted host immune proteins. And then the next step would be doing some in vitro and in vivo experimentation to look for confirmation of your predictions. So what have I shown you here? I've shown you that these viral populations can be used to predict a predicted host-virus interactome. It's also important to point out with this chapter is that these are bioinformatic hypotheses that require biochemical confirmation. So specifically for my system, what I propose, so there's a C-anemone model and a Hydra, and they have some of those classical molecular tools, gene knock-ins, gene knockouts, and some antibodies. So you could have predicted immune interacting proteins, find them in the coral, go test their function in Hydra or C-anemone. And more importantly, this is a rapid pipeline for hypothesis generation that we can apply to any animal out there that we can get viruses out of, which these days is basically any animal. So literally within a matter of a couple weeks, you can go from knowing nothing about a host to have a complete predicted host-virus interactome. And why this is important in the context of understanding evolution is basically the evolution of immunity is based on only 3 of the 30 extant phyla that are out there. So if we're going to chop away at the immunological dark matter of all of the tree of life that's out there, we need some broad tool to apply. And this tool is one I propose. So I've shown you how viruses can of how viruses interact with host genomes and how those can drive evolution. And this work is currently under revised submission at PNAS. So, getting back to the central question of what I showed you in the beginning, we went from this single cell ocean to something that looked like this, an explosion that occurred in only 50 million years or 15 minutes in our 24-hour clock of the history of the Earth. How did this happen? So scientists, we've been trying to figure this out for decades, ever since the first Cambrian fossils were found in the Burgess Shale up in Canada. And so we've come up with some really cool hypotheses, just to go over a couple. So one of these that I really like is called the worm turned and the ocean followed. And this involves a shift of ocean chemistry, specifically sulfide cycling, that was caused by organisms that are that cause bioturbation on the bottom of the ocean, so your worms, your bivalves, etc. And that dramatically altered how sulfide is cycled in the ocean, leading to conditions that led to the Cambrian explosion. And the evidence for this, what we have here is fossilized sediment. This is unmixed anoxic. And you see these nice lines that go across. And then we have this mixed oxic layer, and you see it's just kind of a jumbled mess. So the interesting thing is that Precambrian, before the explosion, we see mostly fossil that looks like this. After the fact, we see everything that looks like that. So it's the sudden appearance of a change of how the chemistry is happening on the bottom of the ocean. Another one involves tectonic shifts and upwelling and the formation of shallow seas. So a paper that just came out in 2014, they have a new hypothesis about when Laurentia, or ancestral North America, separated from Gondwanaland, which included all the other continents. And this caused a flooding from the Pacific Ocean to the ancestral Atlantic to form these shallow seas right here, where we hypothesized the Cambrian explosion occurred. I'd like to point out, too, that this group is from UT Texas. They made sure to put where Texas would be in the ancestral North America. So, getting back to my central question, though, I'm a biologist. That's mostly chemistry. That's mostly geology. What could have been some of the molecular mechanisms that actually led to the Cambrian explosion? So for this specific story, I'm going to tell you the conclusions from the beginning so you know where I'm leading you. So I will show you evidence for a retrovirus infection, a special type called an ERV, which I'll tell you about, that affected these things called genetic regulatory networks. I'll provide you evidence that this occurred during the Precambrian era, and it donated this specific type of promoter element to the last common ancestor of cnidarians, called a long-terminal repeat. This long-terminal repeat is hypothesized, this long-terminal repeat is co-localized with these things called fibroblast growth factor receptors in all cnidarians that are available, and then hypothesized that this donation of the long-terminal element directly affected how cnidarians develop. So, some background so we're all on the same page. So retroviruses, some of you guys have heard of those, probably heard of HIV. So they're this special kind of virus that has an RNA genome, a virally encoded reverse transcriptase, and they have these envelope proteins, the orange guy. So the envelope protein binds to the receptor, the virus genome gets in, reverse transcriptase turns into DNA, gets integrated into the host. From the host genome, where the virus is integrated, produce virus proteins, make new viruses, infect new hosts. However, what can sometimes happen is a complete loss of this orange envelope protein. So when that happens, you have basically a fully functional virus genome that's integrated into the host. However, it doesn't have the proteins necessary to make infectious virus particles. And this has the effect of sequestering the viral genome into the host cell that it's infected. So it can no longer hop around to neighboring cells. However, it can still hop around within the genome that it's infected. So it can now expand within the host genome. And you can imagine, depending on the cell type, this is going to have huge implications on evolutionary trajectories. So if a retrovirus goes endogenous, and that's in some differentiated skin cell type, when that skin cell dies, the virus's progeny dies. That's the end of the line. However, a virus can somehow gain entry to our sperm and egg cells. That virus can potentially be passed on to all future generations. More importantly, the virus's effect on host biology will also be inherited. So just to give you some quick background of what these genomes look like, so this is the envelope protein that I just told you about. This is an integrated retrovirus, and I want to draw your attention to these long-terminal repeats. So these long-terminal repeats, or LTRs, as I'll refer to them for the rest of the talk, they act as, they are involved with integration of the host genome, and also serve as promoters for virus transcription. So long-term repeats have been shown to affect these things called gene regulatory networks, or GRNs, basically how a bunch of proteins talk to each other inside the cell and have some kind of biological output. And just to kind of drive home how dramatic an effect this can have, I have in the center this P53 protein, which many of you have heard, commonly referred to as the guardian of the genome. And it basically talks to everything and is critical to life and death cell decisions on a daily basis. So Wang et al. In 2007 found that 1,500 sites where P53 binds in our genomes is from a long-term repeat donated by a retrovirus that went endogenous. And this puts the virus at the center of host life and death decisions, which you can imagine would be a good, huge advantage from the virus's perspective, controlling how the host lives and dies. So this story, getting to my data, this is a great example of how you can wander in sequence space for 2 years and have no idea where you're going, and then it ends up being kind of cool. So for all of you guys working with your A's and T's and C's and G's, keep trucking, it could work out. Maybe not. So this is kind of the same analysis that I showed you before. I took my viruses, compared them to the whole core proteome through BLASTX, translating those viral gene segments, matching them with host proteins. So that led to the identification of a novel endogenous retrovirus family in the coral genome. So what you're looking at is a 26,000 base pair piece of DNA integrated into the Acropora digitifera genome. And if you'll kind of remember that image I showed you how these genomes are structured, flanked by these long terminal repeats, some domains that are known to be involved with virus replication, helicases versus transcriptases, peptidases, and these are 13 open reading frames within this endogenous retrovirus family, representing open reading frames that are a minimum size of 100 amino acids. So, specifically when I was doing this analysis, I found a portion of this long-term repeat that was extremely conserved in the coral. So I wanted to ask the question, if I took that long-term repeat and went searching for other spots in the coral genome, could I find it? So remember that P53 LTR went to 1,500 different spots. Could I find a similar thing with the coral? So I took my coral viromes, coral proteome, identified that long-term repeat through a different flavor of blast called T-blast-N. I found it in 111 different spots in the coral genome. So at this point, I have there's genome structure, there's predicted domains. I wanted to provide further evidence that the piece of DNA I'm looking at is viral in origin and not host-derived. So integrated viruses can give us some really interesting clues that they actually came from a virus and they're not just part of the host. Two of these are compact genomes and different GC content. So compact genomes. So viruses basically want to use the minimum amount of energy to create a new virus that can be then go on and infect other cells. One of the ways they do this is they minimize their genome size and overlap their open reading frames. And the metric that we can actually measure on this is if we look at their open reading frames or ORFs per base pair of DNA. And then their GC content has also been shown to be different. It's various hypotheses depending on the virus you're looking at, protection from silencing by the host, using rare codons, etc. And so I asked the question, if I go look at all these 111 occurrences in the coral genome, could I look at their ORFs per base pair and GC content and then compare them to random pieces of DNA? So in total, those 111 scaffolds were distributed across 96 different coral genomic scaffolds. So I compared these 96 LTR scaffolds to 96 randomly selected ones. And when we look at GC content here, which is percent GC content, so my long-term repeats, those are the ones that are predicted to be virally in origin compared to the random. We saw that the GC content was significantly higher in those viruses that had the long-term repeat. ORF density, we see something similar, where we have more open reading frames per base pair. That's what this metric is here, in those long-term repeat scaffolds versus those that are just random. So this is providing evidence that these viruses are putting more open reading frames into one piece of DNA. So I've shown you related predicted domains, genome density, GC content, and the general genomic organization that this ERV is actually viral in origin. So getting to this idea that it occurred in the Cambrian. So I asked the question, well, it's all over in the coral, could I find it in other organisms? So I could find it all over, or not all over, but in 6 different ones. And so specifically in all 3 Cnidarian genomes. So in the sea anemone, I could find it in 81 spots, Hydra 541 spots. And then in sea urchins, about 20 or 30, and then in the acorn worms. I'll get into the mechanism for that later. And they're also abundant within the viruses that are isolated from coral and isolated from Hydra, suggesting that this long-term repeat is still hopping around in the viral population. So it's distributed across phyla. So I wanted to see how these long-term repeats might be related to each other phylogenetically. So I did some trees, and then we get something like this, where we see tight clustering based on the source genome. So we see all of our sea urchins here, Hydra genome, coral virome, acorn worm, sea anemone. And what the biological implication of this is that once these LTRs got into these host genomes, they evolved independently, and they were not hopping back and forth between corals to Hydra to sea urchins, etc. So where do we put this in the context of the Precambrian? So we have an ERV infection of the predicted proto-Cnidarian ancestor, so the last common Cnidarian ancestor. And that occurred down here about 700 million years ago. So what you're looking at here, that's millions of years. The Cambrian explosion is all around this time. And the evidence for this is basically that we find it in Hydra, coral, and sea anemone. If I look at any more ancient organisms, I can find no evidence of it based on sequence data. However, I still have to account for the fact that I see it in sea urchins and acorn worms. So this kind of complicates the story a bit. Well, sea urchins and acorn worms shared a last common ancestor called Ambulocaria about 670 million years ago during the Cambrian explosion. So I'm proposing that there was a lateral gene transfer event from one of these Cnidarians, we're not sure which one, into the last common ancestor of sea urchins and acorn worms. So I've shown you the domains, the genome density, distribution. So I told you earlier that long-term repeats are known to be promoter elements. Could I find any evidence of this with my ERV? So to look at this question, I did this analysis called MEME, which basically is an alignment of DNA sequences. It looks for the abundance of specific nucleotides and matches that to known transcription factor binding sites. So when I do that, I get something that looks like this. So what you're looking at here is that is the long-term repeat, here, here, and here. The bigger letter, the more common it is. To do this, I took some representative ones from the coral. And then it was matched to 8 different transcription factor binding sites based on the MEME analysis. So for the immunologists, there's some interesting ones, NFAT-C2, which is involved with T-cell signaling in humans. The one I want to focus on, though, is this PBX1 protein. And so kind of through a side analysis that at first wasn't connected to this project, but then kind of became the nail in the coffin for nailing it, is that these PBX1 proteins are Hox cofactors. And they were originally identified as Hox cofactors that determine central development. Anybody that does development, Hox is one of the most common things you hear about. And these PBX1 proteins are central mediators interacting with Hox. So I wanted to ask the question, I see this PBX1 being donated, predicted site. What about this protein? How is it distributed across evolution? So the cool thing is that it's crazily conserved. So we have here is an alignment from Hydra all the way up to humans of the PBX1 protein. What the letters are doesn't matter. If you see black in the back, that means it's the exact same amino acid. Most importantly, these stars here, those represent the specific residues that are involved with PBX1 binding to their piece of DNA. And that is 100% conserved from Hydra all the way to humans. So we see the appearance of this PBX1 protein apparently in Cnidarians, and I can find absolutely no evidence of any organisms that are more ancient. So we have this appearance of the protein, and then a donation of a putative site by the endogenous retrovirus that I found. So a lot of you guys might be wondering then, well, what genes are affected by this long-term repeat potentially? So what they ended up being are these things called fibroblast growth factor receptors, just showing you some literature. These are well studied actually in Cnidarians, specifically nematostella, which are sea anemones related to corals. And they're basically central development factors of how you go from a sperm and an egg to make a coral or a hydra or a sea anemone. And the cool thing was that I could find my long-term repeat on every single scaffold that expresses these fibroblast growth factor receptors. So what you're looking at here, these numbers indicate their closest homologue to the human fibroblast growth factor receptors. And we see on the coral, they have 5 of them all in one scaffold. Upstream, 1 long-term repeat, another long-term repeat. In Hydra, they've got 1 FGFR, upstream long-term repeat. Sea anemone, they're distributed across 3 scaffolds, and we see it here, here, and here. So in all Cnidarians, I'm finding that this long-term repeat is associated with these proteins that determine animal body plan design. So putting this all together, we have this sudden appearance of a PBX1 transcription factor protein in Cnidarians, which is, the binding sites are essentially 100% conserved from Hydra all the way up to corals. We have the donation of a putative PBX1 binding site within the LTR region, which was donated by this endogenous retrovirus family. And it's known from the literature that PBX1 interacts with fibroblast growth factor receptors in development to create a Cnidarian. So I propose this hypothesis that the long-term repeat I've identified is involved with Cnidarian-specific developmental characteristics. So if we put this in kind of a broader context of evolution, so that P53 story I told you about, so human people have thought about endogenous retroviruses, but there hasn't been put in the context of the Cambrian explosion. So this is a proposed mechanism that could have occurred there using my ancient example as an example that occurred during the Cambrian, and then human literature just kind of further supported. So you have the donation of a promoter element, that long-term repeat by a founder retrovirus that affects the transcription of gene A. That endogenous retrovirus starts to hop around in the host genome, affecting B through G. There's some modulation of a genetic regulatory network that fixates some biological trait. And that has two different outputs. So from our perspective, we see this as evolutionary innovation, new novel biological traits. From the virus's perspective, this is simply just addicting the host to that virus. Once you have a virus that's affected on how to make an arm, you can't get rid of that virus anymore. It's there to stay. So conclusions from chapter 3. So I showed you evidence for an ancient ERV infection based on domains, genome density, GC content, occurred during the Precambrian. Distribution and abundance of this across the different phyla, all Cnidarians, a sea urchin, and a ichthyorn. Through the MEME analysis, it's a predictive promoter element with 8 transcription factors, specifically the PBX1. And this is all co-localized in all Nadarians with these fibroblast growth factor receptors. As with the first chapter, it's important to point out that these are bioinformatic predictions. It's still required, biochemical testing. Basically, that provides evidence that this endogenous retrovirus could have affected, during the Cambrian explosion, how Nadarians began their body plans. So, thank you for bearing with me. Now on to my final chapter. So this is actually kind of going in reverse order. This was the first thing that was published in 2014 in PNAS. And this is looking at the evolution of a specific antiviral response called apoptosis. So what's apoptosis? Referred to it, it's basically programmed cell death. And we have here is just basically like a healthy cell, early apoptosis, late. So during early apoptosis, we see the activation of these enzymes called caspases that basically chew up all the insides of the cells that are then packaged into these things called apoptotic bodies through a process called cellular blebbing. Also, chromatin is condensed and chewed up, and then the final output is, of course, cell death. So from the virus host perspective, apoptosis can kind of be thought of as a classical antiviral response. So a virus gets into a host cell, wants to make more virus. The host senses that virus. The host basically has two choices. It can fight that viral infection, or it can induce apoptosis, kill this cell, and prevent the virus from spreading to other host cells. So I came across these tumor necrosis factor receptors, or TNF receptors, and these are central mediators of this process of apoptosis. And this is a review that was done by Wines and Glennie in 2011 that looked at how many TNF receptors were there across different animals in evolution. In green we have coral, but as a proxy sea anemones, and they're predicted to have one TNF receptor. It's important to point out this is pre-having a coral genome. So this was based more on transcriptomic data and stuff like that, which wasn't a very big database. And the thing I want to point out is you see here 22711, there's this dividing factor, and then all of a sudden we get into vertebrates and we see lots more TNF receptors. So from these data, which was basically kind of based on more of a lack of data at the time, the authors hypothesized that there was a recent explosion of the TNF receptor superfamily that occurred in conjunction with the emergence of the adaptive immune system. So what makes a TNF receptor? So there's three main things that define that protein. So first is this cysteine-rich domain, or CRD. A TNF receptor can have anywhere from 1 to 6. Then are these two loops, the 50S loop and the 90S loop. So being in immunology, there's a bunch of flavors of TNF, a bunch of flavors of TNF receptors, and they don't all talk to each other. So the 50S loop is involved with picking any TNF, while the 90S loop chooses a specific one. So this is kind of general binding, more specific. So I went into the coral system with a newly published genome about a year after I got into the PhD program, and I looked for the cysteine-rich domains in coral. So I didn't find just one TNF receptor, I've actually found 40 of them, and it's the most complex TNF receptor repertoire that's ever been described, more than humans. Interestingly, 11 of them have these death domains, or DDs, and that links the TNF receptor to the apoptotic cascade. And then there's a whole bunch of different flavors. These green things are those cysteine-rich domains, and the different dots are those 50S and 90S loop structures. So they have all these TNF receptors. What about the rest of the components of TNF-induced apoptosis? So corals actually have all of those too. So what you're looking at here, this FAD protein, which docks TNF receptors, leading to the activation eventually of these caspases, which chew up the cell, which I told you about. Then there are these trap proteins, which are adapters, which I told you about a little earlier. Some cool things to point out about corals. Corals have 31 traps, humans only have 6. Corals have 30 caspases, humans only have 12. And my whole dissertation kind of fits in this general body of literature that started a couple years before my dissertation work, that despite their morphological complexity at the molecular level, Cnidarians and other invertebrates can actually be more complex than humans. So they have all these different components. Could they potentially be active? So what would happen if corals were exposed to a human TNF? So the order I'm going to tell you is the order I actually did them. Of course, that's not how the paper is written up. We wrote the paper like we knew what we were doing the whole time. Of course, we didn't. So the first thing we did is we threw TNF on a coral that's in 2D gels. And this is the main figure to come out of it. So in total, there were 92 proteins that were differentially expressed. This is just number of proteins. And I categorized them based on their isoelectric points. So all you need to know is that the smaller value of an isoelectric point, the more negative the protein is. And everything that's in blue basically increase when I add TNF. Everything that's in red, decrease. And so you see this global shift where the reds are going down and the blues are appearing. suggesting that there's a global shift to the proteome, that the whole proteome is becoming more negative. Well, one of the most common methods of single transduction, of course, is putting a phosphate group onto your proteins, which is negative charge. So they have all this TNF stuff. I want to ask the question, could this human TNF actually cause apoptosis in coral? So we just basically went through point by point looking at all the classical ways that everybody else looks at apoptosis. So you have a binding of a TNF to a TNF receptor. You have those apoptotic bodies, which put all the cells components into little nice packaged pieces. Caspases that do all the chewing, and then of course finally cell death. So I isolated some coral cells, added TNF, did some visualization, and some other analysis. So the first one, human TNF binding to coral cells. Excuse me. So we have here our 4 coral cells stained with the nuclear stain DAPI, and added human TNF, did some washes, stained for the human TNF antibody, and every little puncta there, those green dots, those are places where the human TNF is binding to the surface of the coral cell. So we see binding. How about some live cell imaging? So this guy down here is a coral cell, 10 microns, this length right here. This is live confocal microscopy. So I basically added TNF to the cell, watched it, and took pictures. So what's really cool about corals and cnidarians in general, any of you guys use fluorescent probes or any kind of fluorescent protein, you can thank cnidarians for them. They're taken from a sea anemone or from a jellyfish related to corals. And so we don't have to put anything into them to make them really pretty. And this is a coral cell with its endogenous expression of GFP. So I added TNF to this under the microscope, looked to see what happened. So in about 10 minutes, which are these values up here, we see the formation of these structures that look like they could be those blooding structures. And by 60 minutes, they seem to have gotten bigger. So this is one cell looking at one time. What if we look at a different cell type and we look at 200 of them? So here, the different cell type we have in C, these cells are about 14 microns stained with that nuclear stained DAPI. And then in D, this is the classic apoptotic morphology. If you see these dark circles, that's the cytosolic components being packaged to be picked up probably by macrophage. And we saw a shift in the percent of apoptotic cells of this cell type. Percent of cells here, apoptotic in blue, red non-apoptotic, about 15% in the untreated, go to about 70% when you add human TNF. So how about caspase? So you can think of the y-axis as just caspase activity. These are a bunch of controls which don't matter for this purpose. All I want you to concentrate on, the controls work. So we have human TNF, without human TNF, adding human TNF. You add human TNF to coral cells, we see an increase in caspase activity. So finally, the last stage, that eventual cell death, some blind counting, live dead cell staining, number of dead cells here, untreated, human TNF, an increase in the final output of apoptosis, cell death. So putting that all together, we have the binding of the TNF, live cell imaging of a cell undergoing apoptosis, an increased total apoptotic cells, the drivers of the signaling of the apoptotic process, caspases, and finally, cell death. So from here, I conclude that human TNF causes apoptosis in coral. So some of you guys might be wondering about coral TNF. So I didn't tell you, but corals also have 13 TNFs, about the same as humans. What would happen if we put a coral TNF onto a human cell? So to do this, we used a Jurkat cell system. How they're actually QC'd is whether or not they respond to human TNF. And so they have this FAST receptor, which is just a TNF receptor, and this FAST ligand, which is just a flavor of TNF. When you add FAST to Jurkat wild type cells, this FAD protein activates your caspases, you get your apoptosis. We also have this knockout cell type where we have the FAD protein blocked out. And what happens there, you have binding, you have receptor oligomerization, but you have no signaling, no activation of caspases, no apoptosis. So if you added a human TNF on here, what you would expect is you should have more apoptosis here, less apoptosis here because you don't have that FAD signaling protein. So we did that experiment, we did it with coral TNF. And we saw a similar thing, where we have coral TNF down here in the knockout and the wild type. Here we have percent viability. So for that metric, actually the lower percent viability means more death. So in the wild type, we saw more death. In the knockout, we saw less death. So this suggests that not only is coral TNF killing these human cells, it's actually doing it specifically through FAD-mediating signaling. So it's not just killing them randomly, it's doing it in a very specific signaling cascade. So getting to even more central question, where do these TNF receptors come from? So I went back to that cysteine-rich domain, searched around sequence-based other domains of life to see if I could find it. And the cool thing is I could find 6 of them in bacteria. Specifically this unclassified proteobacterium based on the cysteine-rich domains and the 50S and 90S loop. And I think this brings up really interesting questions about where this came from in the first place. Was it a transfer event from a bacteria to a human? Could it be involved with the origin of eukaryotes for the first mitochondria? So why I say that is that all these TNF receptors, none of them have death domains. So none of these, based on the domains we know, can be linked to apoptosis. So could you have had a TNF lateral gene transfer event to the eukaryotic host, a death domain gets popped on by a virus, maybe by something else, and then that fixes it into apoptosis. So putting that in the context of evolution, so if TNF receptors came from bacteria, that puts them at about 2 billion years old. So these guys have been around for a while. And they're still floating in the microbial space, which is also cool. So the cool, elegant experiment, I think I mentioned this one to you before, Roland, would be to take a cell type that doesn't express a TNF receptor. You add the human cysteine-rich domain, death domain, you could just use FAS. And then you switch the domains and add the cysteine-rich domains from bacteria, the death domain from humans. We add TNF, we see, do we still get apoptosis? Is there conservation at the bacterial level too? So what have I shown you? I've shown you that coral TNF causes humans and vice versa. And why this is pretty remarkable is we've basically taken 2 proteins that have not had contact for over 500 million years, and their output is still evolutionarily conserved. Kind of what I mentioned earlier is despite their morphological simplicity, corals are complex at the molecular level, sometimes more complex than us. So kind of chopping away at this anthropomorphic bias that humans are the all-powerful, most interesting and complicated system. Corals have 40 TNF receptors. Which ones are involved with what you just saw is completely unknown. I hypothesized that one of those 11 with the death domains are involved, but we don't know. An interesting study that just came out from Steve Palumbi's lab too, where they looked at TNF receptors in heat-resistant corals. So they found that in corals that were naturally heat-resistant, they had higher expressions of TNF receptors, and they were able to resist more physiological stress. So this could have cool implications into how corals are going to do in the future. So, where have I taken you today? Thank you for bearing with me. So I've, overall purpose, understanding how viruses have driven metazoan evolution from the Cambrian to present day. To do this, I've used corals as a representative metazoan organism. In chapter 2, I showed you that viruses and virus populations can actually be used to predict how host immune systems are structured. I proposed a pipeline that can be applied to virtually any animal out there within a matter of weeks to have a predicted host viral interactome. And that's in preparation for submission. In chapter 3, I showed you how a specific virus that I found affected a protonodarian host genome with implications for, 1, how things could have happened during the Cambrian, and 2, how Nadarians began their body plan development. And that's in a revised submission at PNAS. And then finally, what I just showed you, evolution of this host antiviral response or apoptosis, human TNF on coral, coral TNF on humans, it does the same thing. So with that, that concludes the scientific portion of my doctoral defense. Thank you all for listening. So now, kind of getting into the fun part. So to thank this guy right here, very blunt axe, emerging from the depths of the Arctic ice, Dr. Forrest Rower. Here's Forrest on our trip, holding some freshly brewed coffee. I'm sure this is a scientific article he's looking at, definitely not video games. And then right in front here, you can't see, is where I go running back and forth while I was moving around a lot of different pieces of water. But in all seriousness, when I graduated my undergrad, I had no idea the amazing adventures that I would have during my PhD. Like looking back, I never thought I'd have that in my entire lifetime, much less in 5 years. So I just want to thank you for allowing me to be in your lab. I think the thing that I've learned the most from you is how to ask interesting scientific questions. And I think that's a skill that is going to be really useful for me in the future. So thank you. With my committee members, so Forrest was the chair. Rob and Roland, thank you for letting me rotate in your lab. But as you can see, both of your influences affected the final dissertation output. I had a great time learning all your guys' different techniques. And thank you for serving on my committee. Stephen Hedrick, who couldn't be here today, he provided a lot of helpful insight into the TNF project, specifically that human TNF or coral TNF experiment I showed you. And then Martine Fresqueres, who has been doing some really interesting cell biology, coral cell biology over at Scripps. Then within the Rota Lab, I have to give a big thank you to Dr. Linda Wakely-Kelly. Here we are in Maria. Especially after the last year or two, she's been extremely supportive of my projects, helping with my writing, helping me with finishing off some kind of like thoughts and getting them into the manuscript format. Dr. Katie Baird, who's a postdoc in Dr. Trace Garris's lab now. She was an excellent scientific role model when I first came here. We're still in contact. Aaron, who's been helping me a lot with getting everything you've seen into publishable format, hopefully soon. Yuri, of course, for your immunological insight. So I started this as not an immunologist, and I think somehow I became like a core immunologist or something. And then, of course, Jeremy Barr, for many different things. And then I need to thank, of course, my funding agencies, NSF, which supported me with the Graduate Research Fellowship the first 3 years, ARCS, who supported me the last 2 years. And then the Rota Lab, I've had the honor of working with lots and lots of amazing people over the last 5 years. I don't have time to go through all of them, but it's been great being in the Rota Lab, a very dynamic, awesome place to work, always something new and interesting going on. SDSU, Greg Harris, who's the director of our program, he helped a lot when I served as graduate student rep with Johnson and Vicky. Specifically, a big thank you to Vicky, who, despite being 6 days past due with her baby, she made it out today. So thank you.
No.
Yeah. As well as Lauren, who's not due for another week. And then various faculty I've interacted with in different forms, using reagents, using lab equipment. And then Cameron and Brett helped a lot with some of the fax stuff you saw in the fax facility as well. And then a big thank you to Leslie, Patty, Medora, Renee, who have helped with a lot of the administration. And then special thank you to Gina, who has been a huge help the last 5 years. Forrest and I probably would still be in the Arctic or have never made it there in the first place if it wasn't for her. So thank you very much. And so now I had 3 research expeditions that I was somehow lucky enough to get onto, so I need to thank those people as well. So first, in 2011, I had the chance to go down to Wake Island. So I need to give a big thank you to Dr. Jamie Gove. So during, we left Pearl Harbor a day after the tsunami hit and ended up rescuing people from the Fukushima disaster as well as the tsunami. This is the NOAA guys who just rescued some people who were on an island that was like 3 feet tall when the tsunami hit. So despite all this, we got all of our science done. The interesting thing from this trip actually is this was Jamie's first trip as chief scientist, and the first day he said to me, yeah, I'm going to write my dissertation on this trip. And then he had to do like everything on the whole trip. Then Chip Young, Oliver, and Danny, his nice little pre-picture of Wake. Then in 2011 with Maria, so here's Dr. Jen Smith from Scripps, Andy and Linda, Craig Carlson from UCSB, Craig Nelson in Hawaii, and then the Gump Research Station. We were lucky enough to stay right here for about a month, and here's some of the pretty coral we saw. And then of course the most recent trip, the infamous text that started it all. Here's Forrest texting me about 8:40 on a Tuesday. I didn't pick up. All he said to me was, bring your passport tomorrow. And I asked him why, and he never responded. So 5 months later, I found myself up in the Russian Arctic. I was part of the first non-Russian expedition to Franz Josef Land over 100 years, and I felt so lucky to be a part of it. Some people on the trip, Alan Friedlander, Dave McAloney, who kept us alive. Big thank you to Dr. Enrique Sala, who was the leader of the trip. Dr. Daria Martinova, who came to visit us. Here's that picture Forrest showed you earlier. So the water surrounding us is about -2. And then here's that iceberg that almost killed us. Our token polar bear shot. And with that, I will, finally, most important, my family and friends, especially my boyfriend Billy, who's dealt with me in the last 4 months writing all this up. All you guys who came out to the Ugly Sweater Bar Crawl for the last 4 years, it's been very successful. The course is taking applications for the leader for next year, so get your livers going. Here's my family Forrest. I want to point out while wearing our UCSB shirts. My siblings who have dealt with my nerdiness since I was born. Here's my sisters, same picture. There's my brother right now. And then all my friends that have helped me along the way. So with that, I will take any questions.
You are aware that the tradition of ugly sweater Christmas things is not yet established in France.
That's true, guys. It was definitely in Paris. Yep.
Wow. Wow. Your 700 million years old early retrovirus
Yep.
is unbelievably large
Oh, for retrovirus.
for a retrovirus, which of course can be explained in many, many ways. But most interestingly, if compared to the earliest primate endogenous retrovirus that is so only several years, a million years old rather
Yeah.
than 700,
Yeah.
is very much similar in size to existing retroviruses, which are almost a third of what you showed. But most importantly, there's only one open reading frame, and all several open reading frames without what looks like in your presentation of this 700 million years old virus, many gaps with no open reading frames.
Mhm.
And I suppose you have many theories how to explain that.
So you mean the gap specifically in mind?
Yes, in these 26, I think, kilobases that you showed.
Yeah, yeah. So those could be like with the open reading frames. So specifically the piece of DNA I showed you, it's still it's one piece of DNA, but there are regions of ends, and a lot of those gaps you saw are ends, and so an open reading frame wouldn't come out of that. And also some of the open reading frames end on an end, and so that could actually have been a longer protein that looks like a gap, but is actually just ends. So that's one way that could be explained from that.
So, sorry, I need to ask about this specific virus. I don't remember the picture. Is what you call the three types of proteins, the enzymes, the structural proteins, and the immune response proteins,
Yeah.
are those assembled in the same groups within the genome of the virus, or they are random everywhere?
So do you mean like the accessory proteins?
Yes.
I actually haven't looked at, so within that one I showed you, the specific open reading frames, I haven't matched those ones up to the specific host homo ones yet. So that's something I could look at for sure. That would be good.
Has anybody done Mars? You
No
know?
one's done, yeah, someone's done the variant, and they didn't really find anything.
So have you thought about going back and looking for proto-oncogenes?
No, yeah, that would be really good.
Right, because it seems like the obvious place to look.
Someone in Hawaii did it a little while ago. Yeah. Yep.
In terms of apoptosis in humans, like the blooding, those little vesicles are picked up by macrophages and other cell types. What happens in coral?
So they have Crawley cells too. It's in soft corals they've shown Crawley cells. It's been harder to have like conclusive evidence in corals because of their skeleton. It's really hard to get good pictures. But if sea anemones have them, if other cnidarians have them, corals presumably have them as well. So yeah, you would expect apoptosis should go hand in hand with something to collect those bodies, because if they're just floating around, they're just going to cause inflammation that would have happened. The cells just exploded, right? Yeah.
So you said at the beginning that the viruses enter the gametes. You use sperm and egg as your example.
Yeah.
Do you know an example of a virus in sperm?
In the sperm itself?
Yeah, because I can't get
No, I haven't looked into that,
It's a
but
big size
in terms of
of
like
patient.
retroviruses targeting it, I mean, HIV, you hear all the time how it's always in the sperm. That's one of the most common ways to transmit it, right? And so that's the virus's strategy, that they're already targeting the gonads. So it could be HIV is just trying to go endogenous or something.
But it brings up an interesting point, because you guys the corals can fragment, right? They can do it that way too.
Yeah,
They don't have to just do it
That's
through
true.
gametes.
yeah. Hmm?
Again, I don't remember the name of the protein. PDX1, I believe it was.
Yes.
You show a very specific motif that has 100% homology. PDX1 being a transcription factor, is that correct?
Correct.
Now, interestingly enough, this crazily conserved motif looks like a polyarginine. It's positive, like you have no idea. I don't know if you
Mhm.
realize that.
Yeah, most of them.
Which is interesting. The question is, again, if Forrest teaches me, like he taught you, to ask crazy questions, then the crazy question to ask would be, is a transcription factor, if and when it was transferred, not necessarily from the virus or adapted to the virus, a transcription factor, or maybe a protein that has to traverse membranes, because such a domain will cross membranes and then will allow the protein to traverse to the cell membrane, and then the nuclear membrane that in eukaryotes is necessary to transactivate.
Yeah.
Just, I mean, why out of all the motifs you have this crazy, it looked to me like a lysine arginine motif.
Well, yeah, and so actually the motif was, for the purpose of making that figure, the whole protein's like a 300 amino acid protein, and it's actually extremely conserved across the whole protein. But what I ended up just showing that motif.
Absolute cheated.
Yeah, yeah. Well, I have that piece of data, but like in terms of figures, it's up in the paper now. But it's like,
No,
it's beautiful. It's the whole protein. So it's the motif that's most important that's conserved, and it's extremely conserved of the whole protein. So, yeah.
The coral virions that you compared to the coral proteomes, were those DNA virions or RNA virions, or did they contain both?
Yeah, so they were both. And what's kind of interesting, though, is that this long-term repeat looks like it is being packaged somehow by DNA viruses because it pops up in the DNA. And how that's happening, I have no idea, but virus gene products are known to be packaged by other related viruses, so it could be getting into the viral population that way. But that's a very interesting, subtle part, but all the virologists, it should go off right away, is that a long-term repeat's dealing with retroviruses, which is an RNA virus, but it is popping up in the DNA viruses.
Well, and also the RIG-I pathway was also activated with
Yeah.
the RNA sensing molecule.
Exactly.
So did you see anything specific for DNA sensing?
Nothing popped out. So like the ones that I included in this analysis were the heavy hitters,
Okay.
but there's 2,500 proteins that were within that group. So I could totally go into that.
Yeah.
Yeah, no, that would be good to look at. We could add right to the RIG-1 part.
All right, it's already about 3:30.
Okay.
So let's thank Steve.
Thank you. Thank you.
Go away.
Back to the